about connecting the dots, from discovering fermentation way back to actually editing genes now. It shows how life builds, fuels itself, regulates.
And transmits information. So let's start with the mechanics, the catalytic engines, enzymes.
Right. And there's a great starting point historically. Edward Buchner, 1897.
Ah, yes, the fermentation guy. Before him, the thinking was you needed the whole live yeast cell to make alcohol from sugar.
But Buchner showed that wasn't true. He used extracts, just the juice, basically, from yeast cells.
No cells, but still fermentation.
Exactly. He proved these molecules, later called enzymes, did the work all on their own outside the cell. It was a huge shift. Biology is chemistry, really. Catalyzed chemistry.
And mostly these catalysts are proteins, right? Sometimes massive ones.
Huge, yeah. Molecular weights from maybe 12,000 up to over a million. And their power comes entirely from their specific 3D shape, their native structure.
Lose the shape, lose the function.
Pretty much. And they often need little helpers too. Sometimes it's just an inorganic ion like iron or magnesium. We call those cofactors.
Or sometimes more complex molecules.
Right, coenzymes. These are organic molecules that act like little shuttles carrying chemical groups during the reaction. Think of them as assistants.
There are so many reactions, though. How do scientists keep track?
Well, there's a system, the Enzyme Commission, or EC number. It classifies enzymes based purely on what they do.
So, like, based on the reaction type.
Exactly. Oxidoreductases move electrons, transferases transfer groups, lyases join things together, usually burning ATP to do it. It's illogical. 6, actually 7 now, main classes.
Okay, that makes sense. So the big question, how do they work so fast? What's the fundamental mechanism?
The core idea for any catalyst, enzymes included, is lowering the activation energy. That's the energy hump a reaction needs to get over.
By lowering that hump, the reaction just goes much, much faster.
Astronomical amounts faster. Factors of like 10 to the 17 or even more sometimes. But crucially, they don't change where the reaction ends up the equilibrium, and they aren't used up.
We often hear the lock and key idea. Enzyme is the lock, substrate is the key. Simple.
It's a starting point, but it's too rigid. The reality is more dynamic. It's called the induced fit model.
Induced fit, meaning the enzyme changes.
Yes. When the substrate binds, the enzyme actually shifts its shape slightly. It kind of clamps down, optimizing all those little weak interactions.
Like a handshake, maybe, adapting the grip.
That's a good analogy. Think of hexokinase. It only changes conformation properly when both glucose and its partner, MG-ATP, are bound. It prevents mistakes, increases specificity.
So it's not just waiting, it's actively participating in creating the perfect setup.
Precisely. And this leads to a really deep insight. For maximum catalysis, the enzyme's shape isn't perfectly complementary to the starting substrate.
But to what then?
To the transition state, that super unstable, high energy intermediate state the substrate passes through on its way to becoming product.
Ah, okay. By stabilizing that fleeting state, it makes the whole process easier.
That's where the real catalytic power comes from, stabilizing the transition state.
Okay, we understand the how. Now, how do we measure the how well? That's enzyme kinetics, right? Studying reaction rates.
Yeah, kinetics is all about rates and how they change when you tweak things like substrate concentration.
And you quickly run into this idea of saturation.
Right. If you keep adding substrate, eventually pretty much all the enzyme molecules are busy working on a substrate molecule at any given moment. They form that ES enzyme substrate complex.
So adding even more substrate doesn't speed things up anymore?
Nope. You've hit the maximum velocity, Vmax. The enzyme is working flat out.
And that Vmax helps us define another key term, Km.
The Michaelis constant, Km, it's defined as the substrate concentration, where the reaction rate is exactly half of Vmax.
What does Km tell us, roughly?
It often gives you a feel for the enzyme's affinity for its substrate. A lower Km generally means higher affinity. It takes less substrate to get to half speed.
But is that the best way to compare different enzymes? Or different substrates for the same enzyme?
Not really. For true comparison, you need what's called the specificity constant.
Okay, how do we get that?
First, you figure out kcat, the turnover number. That's basically how many substrate molecules one enzyme molecule can convert into product per second when it's fully saturated, running at Vmax.
So kcat is Vmax divided by the total enzyme concentration.
Exactly. Then the specificity constant is the ratio, kcat divided by km. That number, kcat km, that tells you how efficiently the enzyme converts substrate to product.
And there's a limit to that efficiency.
There is. The ultimate limit is diffusion, just how fast the substrate can physically bump into the enzyme in the solution. Some enzymes are basically perfect. They operate right at that diffusion limit.
Wow. Okay, so they're incredibly fast and efficient. But life isn't always about going full throttle. How are they controlled? Regulated?
Oh, regulation is absolutely crucial. There are several main ways. One big one is allosteric control.
Allosteric meaning other site?
Yep. A regulator molecule binds somewhere other than the active site, causing a shape change that either ramps up or dials down the enzyme's activity.
Then there's covalent modification, like adding or removing groups.
Exactly. Phosphorylation is the classic example. Adding a phosphate group, often by a kinase enzyme, can dramatically switch an enzyme on or off. It's like a molecular light switch.
And sometimes the control's built in from the start, like delaying activation.
That's proteolytic cleavage. Many enzymes, especially digestive ones like trypsin, are made as inactive precursors called zemigens.
So they don't digest the cell that made them.
Right. They need to be sniffed by another enzyme, a protease, to become active, usually only once they reach the right location, like the gut. It's a safety mechanism.
What about stopping them deliberately? Inhibitors.
Inhibitors are key tools, both naturally in the body and as drugs. Broadly, you have reversible and irreversible ones.
Reversible means they can come off again.
Yes. And within reversible, you have different types. Competitive inhibitors look like the substrate and compete for the active site.
So if you just flood the system with real substrate, you can outcompete the inhibitor.
Pretty much. With competitive inhibition, Vmax stays the same, but you need more substrate to reach it, so the apparent Km increases.
Okay, what if the inhibitor binds somewhere else, not the active site?
Then it gets trickier. You could have uncompetitive inhibition where it only binds to the ES complex, or mixed inhibition. A specific type of mixed is non-competitive.
Non-competitive binds elsewhere, doesn't care if substrate is bound or not.
Right, and it changes the enzyme shape so it just doesn't work as well or at all. In this case, adding more substrate doesn't help. Vmax decreases, but Tang might stay the same.
And irreversible. That sounds permanent.
It is. These inhibitors usually bind covalently, permanently modifying the enzyme. Some are called suicide inhibitors because the enzyme actually activates them, leading to its own destruction. Very potent.
Okay, that's a really solid overview of enzymes, the mechanics, speed, and control. Now, enzymes manage energy flow, so let's talk about the fuel and structure molecules. Carbohydrates.
Absolutely. Carbs, the name comes from their typical formula, sort of CH2ON hydrated carbon. They're polyhydroxyaldehydes or ketones, sugars, basically.
We know the basic categories: monosaccharides, the simple sugars like glucose,
Oligosaccharides,